So today I cover this topic like genetic parameter estimation and genotypic correlation and phenotypic correlation and genotypic path analysis and phenotypic path analysis and cluster analysis. Coming to next slides, this is the link already shared. I have group. So suppose this is the genotypic correlation. This is the link. If just click here, you go to direct on Agri Analyze site and just click here, then go to on YouTube and tutorial. This is the dataset link there, and this is the output link. Okay, so these are the here parameters and here the win link, and then go through that. So coming to next slides, this is the RBD, also called the randomized block design. One of the most important design that is the generally used in the agricultural experiments, particularly used in the vegetable breeding, plant breeding. So in this design that controls the fertility variation in one direction only. In this design, the experimental units are divided into the blocks and each blocks are homogeneous in nature and treatments are randomly assigned. So the main objective of the randomized block design, it helps the reduce the experimental error and increase the accuracy and improve the comparison among the treatments. This is the example for randomization of the treatment. Suppose this is the R1, R2, R3, then here the allocation of the treatments in each block. The most important thing is the question is here, how many replication are kept in RBD? If the error degree of the freedom, error degree of freedom is less than more than 12 and equal than 12. So solve this problem. So suppose error degree of freedom is R minus 1 and T minus 1 is more than 12 and equal than 12. Okay. Suppose we have the 10 treatments. We have the 10 treatments means 10. Then R minus 1, then 10 minus 1. Okay. So this value and here replication minus 1 and this value is 9 and here more than and equal to 12. Then R minus 1 and less than equal to 12 divided by the 9. So here R minus 1 then less than and equal to the value is 1.33. Okay. So here the replication is equal to 1.33 plus 1. If the okay, here the R is equal to uh, more than and less than 2.33. It means in RBT design have the 3 replication because more than 2.33 and equal to 2.33. So that's why we keep the 3 replication is must in RBD. Okay. So coming to next slide. So in this type of the analysis divided the total variation into two main parts, variation between varieties and variation within varieties. That is also called the environmental variation. You know that the phenotypic is equal to genotypic plus environmental variance. Okay. So RBD is randomly used in the agricultural research. This is the numerical example of the RBD. The main objective of this numerical is how to make the ANOVA table. Okay. Suppose we have the 5 varieties A, B, C, D, E. Okay. And 4 replication. Okay. So here the sum of the replication first, sum of the replication second, and sum of the replication third, and sum of the replication fourth. This is the grand total GT. So calculation first. Here the correction factor. What is the correction factor? Correction factor is equal to grand total square divided by the total number of observations. Here the total number of observation is 20. Okay. And grand total is 850. So then 856 square divided by the 20. Then here the value of the correction factor. 3663.6.8. This is the correction factor value. This come to the TSS, total sum of square. Here the sum of observation square minus correction factors. Here the sum of square like the 30 square, 35, 42 square till 53 square. Okay, so then minus correction factor. So they are here value of this and minus correction factor. So this the value of the TS, total sum of square. So come to the replication square. So sum of the replication square and number of the treatment minus correction factors. So here is the sum of the replication first, sum of the replication second, third, and fourth. So here the value of the 183,270 divided by the number of treatments is 5. Here the value of the 17.2. This is the RNC. Okay, replication sum square. And again treatment sum of a square. Here the sum square of the treatments and divided by the number of replication. This is the 120 square, 140 square, and 160, 192, and 236. Here value of this. And this is the value of the treatment sum of square 2059.2. So come to the error sum of square, then the here value of the 62.80. So how to make the note table? Suppose the replication and treatments, then error. Then replication R minus 1. Here the replication is 4. 4 minus 1 is equal to 3. And treatment T minus 1. Uh, 5 treatment and 1 is equal to 4. And error is 4 into R minus 1 and 3 minus 1 is means 4 into 3. Here the degree of the freedom. Okay. Then sum of square. Here the replication sum of square. Just put the here value of the replication sum of square 17.2 and just put it here the treatment sum of square 2059.2. Okay. So and error value is the 62.8. And come to the mean sum of square. Then mean sum of square is equal to sum of square divided by the degree of freedom. Okay. If you go to the replication, then R minus 1. And if you go to treatment, then T minus 1. Okay. Then means sum of square divided by the degree of freedom. So 3 and divided by the 4 and divided by the 12. If the divide, then here the value of the mean sum of square. Okay. So this value is mean sum of square. So this is the ANOVA table. So coming to the next slide, this is the ANOVA table. This data obtained after the analysis from the Agri analyze. Here the value of the replication entries. This is the trick, and here the interpretation of the ANOVA table. Okay. How is the p value? If the p value less than 0.05, then significant. If the value of 0.01, then highly significant. If more than 0.05, then non-significant of the p value. So next slides. So this is the mean performance of the each genotypes for character wise, like plant height, number of primary branches, days to first flowering, days to first fruit setting. Okay. So these value are taken from the Excel sheet. This is the mean value and put it here the maximum value, mean value, and standard deviation, period 5% and CV. Come to the next slide. Here the estimation of the genetic parameters like the genotypic variance and phenotypic variance, heritability, genetic advance percentage, PCV, GCV, and ECV. So these are the genetic estimation parameters that is help for the prediction of the selection efficiency and breeding strategy. In these are the also identify the inheritance of the pattern. So coming to the genotypic variance, here the genotypic variance is the total portion of the total variation in a trait. That is the attribute the genetic differences among the individual population. Genotypic variance is the so the heritable factors genes only. Then how much percentage are the transfer in the offspring? Okay. So coming to the phenotypic variance. Phenotypic variance is the total observation variation in the trait, so which include the both genotypic and environmental. You know that the phenotypic is equal to G plus E, means genotype plus environment. Here the variation, genetic factor and environmental factors. Okay, so coming to next slide. So genotypic variance, this is the formula of the M.Sc, mean square of the genotype and mean square of the error and divided by the replication. Suppose we have the value of MSG 150 and MSc value is 30. and divide by the 3 replication. R is the 3. Okay. This is the value 120 and divide by 3, then is equal to 40. Okay. And here this phenotypic formula is genotypic variance. Is the genotypic variance the value of the 40, then 40 and plus MSc value, then 30. MSc value is here, then 30. Then phenotypic variance is 70. Okay. This is the phenotypic variance. If the phenotypic variance is close to if the genotypic variance is close to phenotypic variance, it means low environmental influence. If the phenotypic variance is much higher, then environmental plays the major roles. Coming to next slide, this is the heritability in broad sense. Heritability is the ratio of total variance and upon the phenotypic variance, that is called the heritability. So the formula was suggested by the Verdun Division in 1953. Here the genotypic variance and upon the phenotypic variance, here phenotypic variance is equal to genotypic variance plus environmental variance. It indicates the how much observed the variation in a trait heritable can pass to the next generation. It is not necessary that is the character showing high heritability. If any character showing the high heritability will not also high genetic advance. Okay, that is the Johnson et al. In 1955. So coming to next slide, this is the scale for the heritability also suggested by the Johnson et al. If the low heritability less than 30, then consider as the low heritability, and it means strong environmental influence in that place. Okay. So moderate, if the value of 30 to 60, then consider as a moderate heritability. It means both genetical and environmental effects found. If the more than 60, if the value of more than 60, then consider as the high heritability. It means the mostly genetically controlled. So coming to next slides, this is the genetic advance. Improvement in the mean genotypic value of the selected plant over the base population, that is called the genetic advance. Okay. So it's so the how much gain you can achieve after the selecting the best individuals in next generation. Okay, so here the genetic advance. A is the selection differential at 5% selection intensity, and P, this is the phenotypic standard deviation, and this is the heritability. And what is the selection differential? Selection difference means the difference between mean phenotypic value of the selected plant. This is the mean phenotypic of the selected plant and mean value of the entire population. That is called the selection differential. Okay, it's measure the improvement of the in selection. What is the selection intensity? Selection intensity is the standard version of the selection differential. Okay, that is called the selection intensity. So come to the next slide. This is the scale for the genetic advance. So also suggested the Johnson et al. If the value of less than 10, then consider as the low genetic advance. And if the value of 10 to 20, then consider as a medium genetic advance. If above the 20, then consider as the high genetic advance. Here the interpretation of the genetic advance. If the high genetic advance, okay, here the selection is the very effective due to the additive gene action maybe takes place. So come to the genetic advance. If the value of low genetic advance, here the effective is ineffective the selection due to the non-additive gene action maybe takes place. Come to the genetic advance percentage mean. Here the mean express the genetic gain relative population of the mean and making it easier to compare across the traits. So it helps in the understanding the effectiveness of the selection in a percentage. That is the formula. So G means genetic advance, here the mean and divide by the 100. So this is the genetic advance percent mean. Heritability estimate the indicates the genetic control of the traits, and while the genetic advance percent as mean provide the major expected improvement under the selection. Okay. So come to next. This is the advanced percentage mean also suggested by the Johnson et al. 1955. If the low less than 10%, considered as the low genetic advance. If the moderate 10 to 10, 10 to 20, considered as the moderate genetic advance. And if the more than 20%, then considered as the high genetic advance. Okay, so come to next. At the estimation of the coefficient of the variation. So in plant breeding and variability studies, here the GCV means genotypic coefficient variation and PCV phenotypic coefficient variation is used to measure variability present in a traits. That is the GCV and PCV formula was suggested by the Burton Devon. PCV suppose underroot the phenotypic variance upon the general mean into 100 and GCV underroot genotypic variance upon general mean into 100. So PCV means GCV means it shows the variability due to the genetic factor and PCV shows the genetic plus environmental factor. So if the both value are very close, so it means less environmental influence and the selection will be more effective. Okay, so coming to next slide, here the combined interpretation for the plant breeding. Okay, so if the high value of the GCV and PCV and high value of the heritability and high value of the genetic advance. Okay, so direct selection is very effective due to the here additive gene action may be taking place and genetic variation within the minimal environmental influence. Okay, so if moderate GCV and PCV and high heritability and moderate genetic advance, also here the selection may be very effective due to the moderate genetic control. So if the slightly different GCV and PCV and high heritability and high genetic advance, here the also selection is very effective. So due to the some environmental influence, but the genetic control is still strong. Okay, if the high GCV and high PCV and high heritability, so low genetic advance, here the avoid the selection and use the hybridization and heterosis breeding because here the non-active gene action takes place. Okay, so if high PCV and low GCV, so and low heritability and low genetic advance, here the avoid the direct selection due to the variation mainly due to the environmental factor. So if the moderate GCV and high PCV and moderate high heritability and moderate genetic advance. So also here the careful selection because the both genetic and environmental factor is taken place. So coming to next slide. So this is the estimation of the variability of the component. So that is the table taken from the thesis. So in this table, you just put it here the grand mean value, genotypic variance, phenotypic variance, heritability, genetic advance percentage in GCV percentage and TCV percentage. Okay. So, okay. Now we are going to demonstrate how to analysis the estimation of variability in Agri Analyze. So please everyone log in Agri Analyze. So type here the Agri Analyze. Okay, then go to login. Please log in everyone and we will demonstrate together step by step. If you have not laptop, then write down the step by step. If you have logged in, then let's move ahead.
Yes, sir.
Ah, okay. So you open the file first, genetic estimation parameter of the sample data. Okay. So everyone please open the shared the Excel sheet number 1. So here the right genetic estimation parameter sample data. Okay. Go
Yes, sir.
ahead.
Yes, sir.
Yes.
Yes.
You can see here, this is the genotype. Okay. And this is the replication. And here, this is the trait. TFF means this to first flowering. PH means plant height. So this is the trait given in the short form. Okay. So this is the genotypes G1, G1, G1. Here the replication R1, R2, R3. So suppose you have the 40 genotypes. Okay, here the 40 genotypes, then you must fill the R1, R2, and R3. Okay, it means the arrange your data in the same format shown in the file. Okay, one most important thing, keep in mind that parameter name should be short. Okay, because Agri Analyze application is not accepted. Okay, always keep in mind. So here the code form are the very short form. So these data are entering in Excel sheet. Okay, so Agri Analyze application accepted only CSV format. Okay, so that file, suppose you enter the data in Excel sheet. So how to change the format? Firstly, go to the in file section, then go to export, and here the change file type. Okay, then here the CSV format. Okay, then save it. If you have not CSV format, then Agri Analyze not accepted. Come to again Agri Analyze application. So here the choose the file, upload the CSV format. Okay, choose the GPD, then and here the file successfully uploaded. Okay, then here the select the genotypes and here select the replication and submit it. And here the summary, number of the genotypes 40, replication 33, and total observation 120, and traits 9 traits. Okay, so submit here. Just few seconds wait. This is the complete analysis. Okay, and download the report. So this report will be also automatically sent to the registered email ID. Okay, so click here and to the reports. Okay, this is the interpretation output data. So here ANOVA table of this character, it means dash to first towering. So this is the ANOVA tables, and here the value p value, here the interpretation of the ANOVA tables. Okay, here the standard error mean, standard differences, and CD at 5%, okay, CD at 1%. So here the grand mean, maximum, minimum. Okay, so here this is the environmental variance, phenotypic variance, phenotypic variance, heritability, genotypic coefficient variance means GCV, PCV, ECV. Okay, so genetic advance percentage mean, and this is the mean value. This is the mean value, and here the at par level G20, G16, and here the also interpreted the data. Okay, interpretation of the data for DF for one character is to first flooring. This is the highest value of the object G21, G16, G18, followed by the 20, followed by the G2, followed by G32, followed by 37, 25, 29, 33. So is so here proper interpreted all the data. Okay, and character wise. Okay, okay. So this is the data for interpreted for character wise. You can see here all interpreted data. Okay, you can do also. Okay, if you have any query, please ask anyone at this stage.
Sir, in the site section in the mean values, there is a column of group.
Yes.
So it is also providing us a DMRT data, sir.
Yes. Yeah, this is the DMRT data. DMRT data is always the showing the at par data. right?
Okay.
Yes. So let's move ahead. Okay.
Yes, sir, sure.
Yeah.